Welcome to the Right Science Daily Revision Boost. First of all, a big congratulations to our top 3 scores from day 6, which actually goes to 6 of you. So Aoife in our first place, Fatima and Alicia in second, and then Luke, Izzy and Ravina in third. So well done everyone. Today's daily formula is the force exerted by a spring in newtons is the extension in meters times the spring constant in newtons per meter. Watch out for them using extension in centimeters on this one. Remember to convert to meters. In today's biology blast, we're going to look at mitosis, cell differentiation and stem cells. So mitosis first of all then. When we consider our body cells, they've got to divide in order to repair damaged tissue, to replace worn out cells, and to allow organisms to grow. And the way our body cells do this is through the process of mitosis. Easy way to help you remember, one of your body parts, your toesies, mitosis happens in mitoses. Two things that we need to remember about mitosis: they produce genetically identical daughter cells, and they are clones, which is just a genetically identical cell. One of the things we need to be able to recall is the cell cycle, which is the process of cell growth and division. So I've given you a little diagram on the right there as a reminder. So there are 4 stages to it: the DNA replication, which is your S phase on that diagram, the movement of the chromosomes, cytokinesis, and the growth of the daughter cell. In DNA synthesis, this is where we copy the DNA. So the DNA unzips to create the single-stranded templates, and then 3 bases will pair up with their complementary base pairs. Adenine with thymine, so A with T, and cytosine with guanine, C with G. When it then comes to the process of mitosis, the first thing, the chromosomes line up along the equator, which is the center of the cell. Then they're pulled apart to the opposite poles of the cells by these spindle fibers. Once that's occurred, then a new nucleus forms around the new sets of chromosomes, and then we're going to go on to our last process, which is cytokinesis. In cytokinesis, the cell membrane pinches in and the 2 daughter cells split from each other. Following on from cytokinesis, the cells will grow, and once they're fully grown, the cell cycle starts again. DNA replication, then mitosis, then cytokinesis, then growth again. When we're talking about cell differentiation, we're talking about the conversion of unspecialized cells to become specialized to carry out a particular function. So basically we're making them well adapted for that function. You do need to know some examples for this. So the sperm cell, first of all, has a flagellum to move it, lots of mitochondria to provide energy through respiration, and that structure called the acrosome at the top of the head there, which contains enzymes to break down the egg membrane to allow the sperm to enter. Second specialized cell are the adipose or fat cells. Their whole purpose is to store fat. So the idea behind these is that when we've got them, they can store energy, act as insulation, and act as a protective layer around the organs. And the reason for that is they've got a very small cytoplasm layer around a large fat reservoir that can actually expand to 1,000 times its original size as they fill with fat. Third cell is the red blood cell, which is to transport oxygen around the body. It's got those key adaptations of having a biconcave disc shape to increase the surface area to volume ratio so we get faster diffusion. They contain hemoglobin, which is the protein that binds to oxygen to make oxyhemoglobin, and they don't have a nucleus to make more space inside. The last 2 specialized cells are the ciliated cells and palisade cells. Ciliated cells, their whole purpose is to move the mucus which contains the bacteria and the dirt from places like our airways. And what we find is between those ciliated cells we have goblet cells which make the mucus traps the dirt and the bacteria, and then the cilia sweeps it away from the lungs to the back of your throat where you swallow it down and your stomach acid kills the bacteria. Palisade cells are the plant cells, which are the site of photosynthesis. Key adaptations, they're found near the surface of the leaf. They have lots of chloroplasts to trap that light energy, and they've got a regular shape so they can be packed close together. When we talk about a stem cell, this is the undifferentiated cell. So they can divide by mitosis and then differentiate into any type of specialized cell that we've already looked at. We've got two types of stem cell, adult stem cells and embryonic stem cells. The embryonic stem cells are found in embryos, as the name suggests. They divide by mitosis and can differentiate into all cell types. Whereas an adult stem cell is found in various tissues around the body, so brain, bone marrow, skin, liver, and they can differentiate into some cell types, and they're used to repair damage. Key difference there is embryonic, all cell types, adult, only some. When we're thinking about plants, their stem cells are referred to as meristems. Now, the plants will grow throughout their whole lives in certain areas, the root tips, the shoot tips, and the buds. Key thing about our meristem cells, they're small, they've got very thin walls, small vacuoles, and no chloroplasts. Just a quick reminder of our daily formula, which is the force exerted by a spring in newtons is the extension in meters times the spring constant in newtons per meter. Today's chemistry check is on polymers, the periodic table, and metals. When we're talking about a polymer, they are made from many alkene monomers being joined together. And in order to do this, we need high pressure and a catalyst. So when we've got this reaction, it's called addition polymerization. We're just joining lots of alkene monomers together. When it comes to writing the name for our polymer, you start off with whatever the monomer is, stick it in brackets, and write the word poly in front. If they give you the polymer name and ask you to write the monomer, take the poly away and just write the word in brackets. Same kind of idea with our displayed formula at the bottom. On the left is the monomer, on the right is the polymer. So we lose the double bracket in the middle, stick the little arms out the side, put brackets and a little N. We've got two types of polymer we need to know about. Thermo-softening polymers, which are just these little lines representing our polymer chains on the left, and the thermosetting polymer, where they're joined by these cross-links. So cross-links are covalent bonds which join those polymer molecules, and that makes them harder to separate. The non-cross-link polymers are easy to separate as they've only got weak intermolecular forces which are easily overcome. When we think about metals, we need to remember that they're all solid at room temperature except mercury, which is a liquid. The atoms are arranged in a giant metallic lattice so that they're arranged in a regular pattern, as you can see on the right. So that in our metals, the electrons leave the outer shell of the metal atom to form a positive metal ion and delocalized electrons. Then those electrons are free to move through the structure of the metal, and what we find are that we get these strong electrostatic forces of attraction between those delocalized electrons and the positive metal ions, and that's our metallic bond. Last part of our chemistry check is the periodic table. So Mendelev's periodic table formed the basis for what we still use today, and he considered a range of evidence in constructing it, including the atomic weights of the known elements, knowledge of the chemical reactions of different elements, and knowledge of physical properties. He then arranged those elements into order of atomic weight and into groups. So he actually left spaces where he thought an element hadn't been discovered yet that should go in there. Following on from Mendelev's work, in 1913 Moseley discovered the atomic number was the number of protons in the nucleus, and this then proved that what Mendeleev had done was exactly right. In 1894 we had another contributor to the periodic table, which is Ramsay, as he discovered argon. Now this was an element nobody had predicted the existence of, and then he discovered helium. By 1898 three more elements were discovered, neon, krypton and xenon, and he suggested to Mendeleev that these all formed a new group that should be placed to the right of group 7, and they're known as our noble gases today. The reason they haven't been discovered? They're just really unreactive. So when it comes to our modern periodic table, we need to remember a few facts. Elements are arranged in order of increasing atomic number. The atomic number is the number of protons in an atom. The number of electrons is the same as the number of protons in an atom because they're neutral. Electronic structure is determined by your electron number, and the electronic structure determines the chemical properties. Quick reminder of today's daily formula. The force exerted by a spring in newtons is the extension in meters times the spring constant in newtons per meter. Today's physics prep, we're going to be looking at magnetism. First fact, like poles repel, opposite poles attract. Now, when we're looking at these diagrams, then we will draw these field lines, and they represent magnetic flux. So the number of field lines passing through a particular area is called the magnetic flux density or magnetic field strength. Remember that magnets will move in the direction that makes the field lines shorten. So we've got two types of magnet, the permanent magnet, which is magnetic all the time, and an induced magnet, which is where we will only have a magnetic material becoming magnetic when it's in a magnetic field. We can model magnetism using the domain model of magnetism, and that tells us that a permanent magnet is made up of many small magnetic regions called domains that are all lined up. If we think about a material that might be magnetic, so iron or steel, they have regions that are not lined up, but when we place it in a magnetic field, they do line up. So that when we then remove that material from the magnetic field, then they may return to their original direction, or potentially they could stay lined up. When we're using a compass, then it's actually pointing towards the magnetic North Pole, which is different to the North Pole of the Earth. So that what we find is the reason our compass does this is because the Earth behaves as if we've got a giant bar magnet at the centre. And the reason behind this is that scientists think the Earth's magnetic field is caused by convection currents in the molten iron core of the Earth. If we think about what happens when a current flows through a wire, then we produce a magnetic field around it. So if we think about just a single strip of wire first of all, then what we end up with are a series of concentric circles as the shape of our magnetic field, as you can see in the diagram. And if you want to work out the direction that they're going in, use your right hand for this. Have your sort of hands around like you're doing a thumbs up. Your thumb points the direction the current's moving, and your fingers point the direction that the magnetic field is moving in. Two key things that our magnetic flux strength will depend on: the magnitude of the current, so a bigger current means a stronger magnetic flux, and the distance from the wire. The closer it is, the stronger it is. And don't forget, when we're talking about the strength of magnetic fields, we're using the units Teslas with a capital T. If we consider what happens with a loop of wire, then the magnetic field in the centre of that loop is a straight line. Around the actual edges of the loop, you can see we get those concentric circles still. If we place a magnetic material in the core of our coil of wire, then we create an induced magnet when the current flows. For those of you doing the foundation tier, you can head on over to Socrative to do the quiz now, because the remainder is only for the higher tier students. So for those of you doing the higher tier, if a current carrying wire is placed inside another magnetic field, then those two magnetic fields will interact. So they push on each other and create a force on the wire. And that force is always at right angles to the magnetic field. So when two fields are in the same direction, then they add up, and if two fields are in the opposite direction, then they cancel each other out. So if we consider putting our current carrying wire into a magnetic field, then we create a catapult field. So we've got fewer stretch lines below than we do above. As a result of that, the wire is going to move down so that the field lines straighten, because that shortens the field lines. If you want to know what direction the wire is going to move, we're going to use our left hand this time, so do make sure you know the difference between your left and right. If in doubt, just hold them out with your thumb sticking out and your left hand makes an L shape. Then we're going to use Fleming's left hand rule. So stick your fingers out like a bit of a weird gun, and then your thumb is the movement of the wire. Your first finger, or your index finger, points in the direction of the field from north to south, and your second finger is your current. So that points from positive to negative. So you might do some weird movements in the exam. Hopefully your science teachers have told the invigilators you're not using some kind of weird code, but it means you can sort of adjust your hand, work out what direction the field and the current are in, and therefore know what direction it will move. If we think about how we can apply this to motors, If we take a loop of wire and place that in a magnetic field, then we're going to have a force generated on each side of the loop. And as a result of that, as the current flows through it, it will turn. Because you can see in that middle diagram, you get two catapult fields, one side going up, other side going down, so it spins. In order to make that coil spin continuously, then the current needs to move in on one side and out on the other constantly. So we achieve that through the use of a split ring commutator, which I've given you on the right hand side of the diagrams there. The key thing here is that we've got a little gap between the two sides, so that it allows the current to flow the same way from the battery, but the coil will change halves as it spins. So the force on the left of the coil is always up in this case, and the force of the right on the coil is always down. Last thing to consider, how can we change the speed of a motor? We can change the magnitude of the current, the strength of the magnetic field, the number of coils in the wire, or the length of the coil. Just go careful on these questions that if it says how can you increase the speed of the motor, don't just say change the size of the current, say increase, because you've got to be specific to link it to what they're asking. Final reminder of our daily formula then, the force exerted by a spring in newtons is the extension in meters times the spring constant in newtons per meter. Now we've had a quick recap, then head on over to socrative.com and test your knowledge on the topics from today. The room name you need to enter is Right2662. Good luck! If you're unsure on any of the topics we've covered in today's Daily Booster, then head on over to the playlists page and pull up the more in-depth videos. So for the biology section, it's B2, chemistry is C2, and the physics, a little bit weird, if you're doing combined science, it's the P3 playlist. If you're doing the separate science of physics, then it's the P4 playlist.